Okay. Am I audible? Is my screen visible? Are you able to hear me? Okay. Thank you so much. Okay, we'll start the class. We'll see if someone else joins. They can continue from where they're joining. Okay. So thank you for joining today's session. Today's session is on bioenergetics, and we'll also begin with glycolysis. Okay. So um, let's begin. So, in today's session, we will cover the bioenergetics portion, where we'll look into different concepts of bioenergetics and what all different aspects are incorporated into this. And we also have some numericals that are that you already have must have seen in the this week's course notes, and we'll also do it in this class so that you get more practice of it. And then we go to glycolysis, and we just touch upon this and just see the first phase of glycolysis. In the next class, which is in the next week, we will do glycolysis in much more details. For today, we'll only begin and just see how glycolysis, the first 5 steps of glycolysis, take place. Okay, so let's start. So today we are going to mainly focus on bioenergetics. So bioenergetics, as you would have seen in the lecture notes as well, is basically the quantitative study of the energy transduction that occur in living cells and the nature and function of the chemical processes underlying these transductions. So as the name suggests, bioenergetics. So you're looking into the energetics. Energetics means anything that is to be done with the energy transduction, anything that is taking part in transferring energy or conducting energy, that is energy transduction process. So anything that is doing such kind of transduction in a biological system, we're going to look into that, and we're going to quantify that energy transduction. Okay, and by quantification what we mean is how much of this energy transduction is taking place in terms of their energy, in terms of their um, energetic expense, and what type, so nature, and what is the function of the chemical process which underlie these transactions, right? And like any other system, in biological systems also energetics follows the two laws of thermodynamics, the first law and the second law. So first law, as we know, it says that the energy is constant and energy is neither created nor destroyed, but it is only transferred from one form to another. And the second law of thermodynamics talks about the tendency of the universe to expand, so to increase its entropy, right? Now in living beings, if you see living organisms, the cells are quite structured, they are well defined structured. So you would expect that this is already in a defined structure, in a well organized manner. Then why are we saying that even living organisms are following the second law of thermodynamics? Because second law of thermodynamics means more chaos, more distortedness. So but cells, if you see in general, they are quite well organized. So if that's the case, why are we calling this as a second law, following the second law of thermodynamics? Well, we do so because we are talking about universe and not the system only. So if you look at the law says the universe. Just a second. Yeah, so if you look at the law says that the universe tends to expand. The universe has a tendency to increase the entropy. So if you consider living organisms or living cells, what is the universe? It is the system is the cell. The universe will incorporate its interaction with the outside surrounding or the surrounding the cells. So that surrounding and the cell together make up the universe. And we know that in living organism will constantly exchanging some molecules, some of the components with the surrounding. So because we are in an interaction always with the surrounding, that's how we are always, even in the living cells are also towards the increase in distortedness, increase in the chaos. So they also follow the second law of thermodynamics. First law of thermodynamics makes sense. Energy is there. Energy can neither be created, neither be destroyed. It's only transferred from one form or another. So that is understandable. Now, the next concept that we all should know is the Gibbs free energy. So Gibbs free energy is given by, let's look at the first, let's look at the left side. So it's given by G. So delta G, that is change in the Gibbs free energy, is given by delta H minus T delta S. So what is G here? G is basically Gibbs free energy. What does it mean? So it basically talks about the amount of energy which is capable of doing work during a reaction. Okay. And that is at a constant temperature and a constant pressure. Now, when a reaction, in general, when a reaction is taking place, if it proceeds with the release of free energy, so what does it mean? That the system changes so as to possess less free energy. So the delta G will then have a negative value and the reaction is that exergonic. We already know this, right? That your delta G, if it is negative, the reaction is spontaneous. It tries to release free energy. It tries to reduce the free energy, right? So your delta G, as it says here, if it is negative, it is spontaneous. If it is positive, which is greater than zero, then it is non-spontaneous. And if it is zero, then it is at equilibrium, right? Now, how do you come to this delta G being negative? The factors which are incorporated into this are delta H minus T delta S. What is H? H is enthalpy. Enthalpy is basically the heat content of the reacting system. So whatever reactants and whatever products you are seeing, it is the heat content of those products and reactants. Now, where does this enthalpy come from? The enthalpy is basically reflecting the number and the kind of chemical bonds that are present in these components. So the kind of bonds which is in there in the reactants and it is in there in the products, right? So when a chemical reaction, if it is releasing heat, it is exothermic. So it is releasing heat. It is kind of giving out heat to the surrounding. So the heat content of the product is less than that of the reactant, right? So listen to this once again. If your reaction is exothermic, if your chemical reaction release heat, then the heat content of your product, which is on the right side, reactant giving rise to product, is less than that of the reactants, and therefore delta H has a negative value, right? Conversely, if the reacting system take up heat from the surrounding, which are endothermic, then your delta H will have a positive value. So you have a delta H which works according to what is the heat content. Are you releasing heat while you're undergoing chemical reaction? Are you taking in heat, right? T, as we know, is temperature. Delta S, S is entropy. So entropy, as I have already mentioned so many times right now, it is basically about the randomness or disorder or chaos in the system, right? So when the products of a reaction are less complex and more disordered than the reactants, the reaction is said to have a gain in entropy. So listen to this. If your product is more disordered, has more chaos, it's not structured, it's not in ordered form, there is a gain in entropy. Entropy inherently is the tendency of the system to be in a chaos or in disordered state. So if your product is more disordered than the reactant, then you have gain in entropy, right? So all these, just a second. Yeah, all these taken together, your delta G becomes H minus T delta S, right? Your gain in the entropy. So your this constant on the T delta S becomes negative. As T delta S becomes negative, your minus delta G also becomes negative, and that makes it more spontaneous, right? So that's one way of writing or one way of looking into the relation between enthalpy, entropy, and Gibbs free energy. The other way is to look at the delta G, which is given by minus RT ln K eq or K equilibrium. Now, what is this? So let's say this is if you, so there's one very minute difference, but you should realize it when you are looking in biochemistry. So here, if you look into this delta G, it is delta G prime value, right? This delta G naught, you will see G naught, G naught, E naught, these you will see in chemistries also. Those are standard equation. This right now here is a standard equation, yes. But a standard equation in general, if you look in chemistry, it's basically when there is pH 0 and the concentration is 1, right? For water, if we will start with water, in chemistry, we will see if we are defining standard state, it's at pH 0 or at molecular concentration of 1 molar. That's okay. But in a living system, in biological systems, you will not see pH 0. What you will see is pH 7. So the standard state, which is defined in biochemistry, is referenced at pH 7, and if we are talking about water, it is at 55 molar. We have already done it in the previous classes. 55 molar water concentration is the standard condition, pH being 7, right? So because our in biochemistry, the standard state is a bit different than chemistry, these have this prime, delta G prime naught, is called transformed delta G. This minute difference, even though it's minute, but you should understand that this difference exists and you should realize the importance of this. You should keep this in mind, right? So when we talk about that, we're basically talking about delta G prime naught is basically minus RT ln Ke. R, as we know, gas constant, T is temperature, ln Ke. K equilibrium is basically talking about the concentration of the product to the reactant, right? Now, this is standard equation for the initial concentration of each component is 1 molar. That is satisfied. Remember, now I was talking about water when taken as a reference. But any other product and reactant, the standard equation is basically at 1 molar, pH 7, temperature 25, and pressure is 101.3 kilopascal. Or if you have something which involves, let's say, Mg2 plus, then that is 1 millimolar, right? Concentration of H plus at that condition is 1 millimolar, not 1 molar for H plus. So these are minute differences that you should remember. Okay. Now, coming back to it. So this delta prime G naught is a constant because it is standard. It's a given condition, standard condition. So your delta G naught is actually a constant value. But in cells, neither do you have everything at 1 molar, or your conditions might change. Because your conditions change in living cells, your delta G is not, it deviates from the standard. That's what it's written here. So the actual free energy change, which is delta G, not G naught. G naught is standard. So G is a function of the reactant and the product concentration and the temperature prevailing during the reaction. So temperature, because everything else here, T, and this factor here, delta G naught, is constant. So this is constant, and this this R is always constant. T is what can change, and these is what can change. So when you need to find out the delta G, that is the formula for figuring out delta G, what you are doing is delta G is equal to the standard transformed delta G plus RT ln product by reactant. Now, if you say at equilibrium, when you know that delta G is not changing, delta G is 0, then the equation that we did in the previous slide comes to form. Delta G is equal to minus RT ln Ke, which is at equilibrium. And in equilibrium at standard condition, that's how you connect all the dots. But if you are to calculate the delta G in a living system or in a biological system, this formula here is what you need to use. Okay? Right. Now, so delta G is basically then the difference between free energy content of the product and the free energy content of the reactants under standard condition. We have already understood that. We already know when delta G is negative, reaction takes place spontaneously. When delta G is positive, reaction is non-spontaneous, right? So when delta G is negative, what actually happens in terms of products and reactants, let's try to understand it a bit better. So when delta G is negative, the products contain less free energy than the reactants, and the reaction will proceed spontaneously under standard conditions, right? All chemical reactions will definitely tend to go in the direction that results in the decrease in this free energy of the system. That's what we have been talking about, right? While a positive value of delta G will mean that the products have more free energy than the reactant, and this reaction will tend to go in the reverse direction if we start at standard conditions. Okay. Now, so this is delta G. All the chemical equation or chemical reaction have their own standard delta G naught, and that given your cellular condition or your conditions when it is in present in whatever present intracellular will deviate from delta G naught. Delta G naught is your standard. Whatever is delta G, that is your uh, given your current conditions. Delta G, if it is negative, delta G, if it is negative, then the reaction will take place spontaneously. If it is not, it will take place in the reverse reaction. That's clear. But what is what happens if when you look at delta G naught, okay, delta G naught is a standard condition. If delta G naught is positive, what happens there? Does the delta G, but so delta G naught is and the standard condition means if delta G naught is negative, that means if delta G naught is higher, that particular non-spontaneous. But what happens is if you have a reaction with positive delta G naught, that can also go in the forward direction if your delta G is negative, right? Now, what can happen here? So let's look into this equation once again. So you have this delta G and this equation here, which takes into the standard delta G naught, so standard free energy change plus these RT and the mass action ratio. Now, this is positive, and but this should be negative, right? Only then can the reaction take place. So if this is positive, this equation has to become negative, and such negative that this is greater than this value of delta G naught. So if your products and reactants are in such a concentration that it renders this whole value to be negative, then your delta G can go forward even if at standard condition that reaction is unfavorable. So that's how concentration determines if the reaction takes place or not, even if your standard says it cannot take place. So concentration of the reactants under the intracellular condition is what all drives the can drive the energy change and can drive the reactions, right? Okay, so all this discussion brings us to few of numericals so that you get the concept much better. So let's look at the first numerical here, right? So you have a final equilibrium mixture at 25 degrees centigrade, so temperature is given, pH is given, so H plus concentration is also given and pH is also given. Then the reaction is what we are looking is basically glucose 1 phosphate becoming glucose 6 phosphate, right? The concentration of product and reactant is also given to you. 1 millimolar of your reactant, 19 millimolar of your product. So you are supposed to calculate the equilibrium constant, which is K equilibrium, and then the standard free energy change, which is delta G. You have the R value given here. Can you calculate both these value and let me know if you can get to it? So you basically have been given your, so if I go back to the formula here, we basically have been given here almost everything. So you need to calculate this component and you need to calculate this component, right? So can you let me know which value will it be? Okay. So K is 19 and what is delta G? Mm, yes. Okay. Can you give me answer in kilojoule and also check your value once again, like unit? Yes. But are you sure about the value, sorry, the unit of delta G? Is it just kilojoule? Okay, maybe I'll give you the answer and then you can check, right? So you're correct. It's minus 7.3 rounded off kilojoule, but it's kilojoule per mole. And I'm guessing this is what you did. So K equilibrium, as we know, it is basically product to reactants concentration ratio. So we have been given here 19, and we know that the reactant is 1, so 19 by 1 is 19. So you got the equilibrium constant correct. And then the standard free energy change, which is minus RT ln Ke. So RT value is already given, J mole into K, 298 Kelvin, which is temperature, and ln of 19. So when you do that, and you report the answer in kilojoule, the answer will be minus 7.3 kilojoule per mole. So kilojoule per mole or joule per mole is the unit for free energy change. So be mindful of the unit when you are writing the answer. But you got the answer correct, so that was great. Okay. All right. The next part is, so you have the Ke. Uh, just a second. Yeah. So you have the Ke already given to you, and delta G is given to you. What is the value of the equilibrium constant here? So you have your one entire value given, and you need to find out the Kex value for me. Your delta G is given. You know R. You also know the T here, and you need to give me the Keq value. Substitute the values in the equation. You will get to see what exactly you are trying to do, and then do that equation, solve the equation, and let me know the answer. So I do hope that you know that this is the formula that is supposed to be used, right? You have your Ke value. This is told that you need to find it out. T is given to you. R you know you are given also, and delta G also is given to you. So you need to just calculate what is Ke, and for that you need to do a transformation, which you should be able to understand the moment you see it in the equation. Point 9862. Mm, okay. All right, let me give you a hint. Let's see if you have done the same, if you want to change the answer. So you know your delta G, which is 35 minus 35. You know, which is equal to minus R. So R you know is given to you already. And you have ln, Ke and T. So you have 37 degrees centigrade, convert that to Kelvin. So what happens is you can take ln Ke on your left side and do the remaining calculation, that is delta G divided by minus RT on your right side. Okay. You get a value which is ln Ke. You need to do the natural log or natural, so you can convert it to natural log and get the K equilibrium value. Is that what you did? Yeah, I understood. So do it once again, and then we can look at the answers. Ten to the power 6 for. Okay, you giving answer. Yeah, I understood. Okay. All right. Maybe I will show, and then you can see if you have any doubt in the answer or if there's step that we are showing. So the answer to the K equilibrium constant is 7.8 into 10 to the power 5. Don't get overwhelmed. It's fine. We'll see how it's happening. So I told you this is the equation that we are using. Delta G naught is equal to minus RT ln Ke. So I told you ln K can be taken on the left side. Delta G naught divided by minus RT. That much is fine. Let's just substitute the values. Delta G naught is minus 35. Remember, joule, it's kilojoule. So let's transfer, either you can convert joule to kilojoule or kilojoule to joule. Because multiplying is easier, so let's convert kilojoule to joule. So minus 35,000 divided by minus 8.3143 as given here into 310.5, which is 37 degree centigrade into Kelvin. You get a value. So your Ke is basically, ln Ke is basically 13.5732. To get K, either you can do exponential or you can simply convert log K and then do lateral log to get the value. And did it make sense? Did you get confused? I'm guessing that did you got confused in the step of converting and bringing them to the same unit? Yeah. So in my all classes, I always mention, please look at the unit, please look at the answers. Your unit should always be similar. Do this. So that's why this practice was important. Yeah. So if you do your units, or your units should always be in the same, like they should always be similar. uh, the trick is that if it's kilojoule or anything in kilo and anything in joule, it's easier for you to get the bigger unit to a smaller unit because it's multiplying then by dividing, but in certain cases even division works better. But the idea here is to get all the units in the same or all the uh, values into the same units. Okay, and yes, if that had happened, you would have gotten the answer. Okay, right. All right. Now you have some statements here based on whatever we have discussed so far, and you need to tell me which one of these are true, right? So you can read these 10 statements and let me know which one's true and which one's false. C only wrong. Okay. Okay. The rest of the statements are true. All right. I'll wait for the other person to answer and then we will look into the solution for this question. Okay. Uh, Only A, is it the answer? Let me give the answer then. That's it. Okay, you're writing more. Okay, I will wait. So A, B. D. E. G. H. I. J. So whichever you feel is correct. Okay. All right. You can check if you are correct about the rest three also. Okay. I will let you first see and then I will tell you what's exactly happening. So I'm looking at all these statements which are true. The true statements are B, C, D, E, G, H, J. Okay. Did you write more? No. Okay. So A, I think both of you said A is true. That's not correct. I'll tell you why. And we have already looked into this why. Wait. I will show it to you and then I will maybe explain it to you. Where did it go? 101.3 kilopascal. 25 degree is correct. So what is the question saying? Sorry. The question says 1 molar concentration species for standard condition, correct. 25-degree, correct. But 1 Pascal pressure? No. If 1, then 1 atmospheric pressure, otherwise 101.3 kilopascal pressure. So unit to be clear. Okay, no problem. Second, I guess you said all correct. All of both of you said correct, which is correct. Delta G naught is for a reaction is basically change in free energy under standard conditions and pH 7. C, you said wrong, and I think you also said it's wrong. C is enthalpy is closely related to the nature of the atoms and bond present in the compound, and that was correct, wasn't it? We discussed delta G is free energy, which is amount of work to be available to be done. Delta H is enthalpy. Enthalpy is intrinsic to the product and the reactant, and that is determined by the nature of the atoms and the chemical bonds that the compound has. That determines what is the amount of heat in that component. So enthalpy is related to the nature of the atoms and the bonds, right? D, I think you said correct, both of you, which is correct. Entropy is related to the disorder. We have done this enough time. E, again, you all, both of you said correct. Gives free energy, the amount of energy available to do work. That's correct. F, all the energy obtained by a change in enthalpy is available as Gibbs free energy. That is incorrect, because you also lose some in entropy while changing your while undergoing a change in entropy. Some of the energy is lost. So not entire all the energy that you are obtaining by changing the enthalpy is not available as free energy. Some go out in the form of entropy, right? G, an exothermic with positive energy will be a positive entropy will change will be a spontaneous. That's correct. And then H, as we already know, right? We have done this formula here. Delta G is equal to RT ln KE. So we know that it's a logarithmic function. I, while most biochemical obey the thermodynamics laws, there are some exception that is incorrect. There is no exception to any biochemical reaction. All biochemical reaction follow the law of thermodynamics, which are the two that we have discussed. And lastly, which I think you got correct, which is when fully oxidized, palmitic acid generates more ATP per unit weight than glucose. This we did in the previous session, because we know that fatty acids are more reduced than glucose, and that's why they generate more ATP. But that's okay. That was the answer to this. But what you need to remember, once again, I would like to highlight is the conditions for standard. Whenever we are talking about standard condition, all the conditions you should remember. And the other thing that we missed out was, if you just read it once again, that would be easier for you to know. Okay. All right. So that was the first part of bioenergetics, where we looked into the delta G and how is it relating to enthalpy and entropy and the concentration of reactants and products. Now let's look into ATP. So there is not much thing that we are doing in ATP. We're just going to see how is it working, right? So energy currency or ATP is used as energy currency, that we know pretty much know, and we're going to do it over and over in the next few sessions in many different pathways, where we'll see that it is using ATP to undergo a chemical reaction, and we'll also see how ATP is generated during after the Krebs cycle. We'll see all of this. So we will see that ATP is the energy currency. So what is special about ATP as an energy currency? It links the catabolism and anabolism. So we remember, right? Anabolism is a reaction where we are looking towards the production of a compound. Catabolism is where we are looking into the breaking down of the compound. So when you look into heterotrophic cells, which are basically obtaining free energy in the chemical form, so they are doing that they're obtaining by catabolism. We know this already. We know glucose is taken up and glucose is broken down into gain energy, and that whatever energy is gained from there is used to make ATP. So ATP is generated by the intake of glucose, or when there is breakdown of glucose. That much we know. Now this is how ATP is formed. So that is anabolism. Now, what or catabolism reaction that gives ATP. Now, what does ATP help in do? ATP donates the chemical energy that it has to several endergonic processes such as synthesis of metabolic intermediates or macromolecules. And what it does is ATP has energy stored, so it gives its chemical energy to the endergonic, whichever processes that requires energy. So ATP gives that energy, energy currency is being used, and that reaction goes forward. ATP is hydrolyzed to give ADP, right? So this donation of ATP generally involves a covalent participation in the reaction that is to be driven. And generally in the cells, ATP, whichever reaction we will see ATP is being talked about, remember most and often ATP is in the Mg2 plus Mg-ATP2 minus state. Basically, there's an Mg which is bound to the ATP. So that's why, not here right now, but later we'll see how it, later in the next sessions, we will see that it matters to the overall energy change, differed how that differs from just ATP being an energy currency, and then how Mg2 plus being covalently bound to ATP makes the energy current, the overall energy different. But what you should remember for your understanding sake is that ATP in intracellular condition is mostly found in bound to Mg2 plus. Okay, now, but what is even more important that you should remember is, I very simply said, ATP gives ADP. So adenosine triphosphate becomes adenosine diphosphate, and it gives out a phosphate, and that phosphate is what is driving the chemical reaction. And it looks like this is a one sample step reaction, and it is just by hydrolysis of ATP. That is, hydrolysis means using water and breaking down the ATP into ADP and phosphate, that this reaction is going on. And there is no other step taking place. But what actually happens is that the ATP is actually covalent, non-covalently, or how you can say that it is bound to the reactant while it is driving the energy. This is what we mean for understanding purpose. This is what. So the first half, the A section, is what you will generally see written. So let's say glutamate is to be converted to glutamine. So you have to add this ammonia group here, amine group you have to add to the glutamate. You know this, right? And you will see for that, the energy that drives this reaction is by using ATP hydrolysis. So ATP is hydrolyzed to give you adenosine diphosphate, and one phosphate is removed. That's what we will see. And it will look like, oh, it is a hydrolysis. So this one breaks down, and while breaking, whatever energy is generated, that energy is being driving the reaction where ammonium is getting bound to the glutamate. But that's not what happens, and this is what you should remember. Energy donation by ATP is not just simple hydrolysis, it is by group transfer. And we know this terminology. If you look into it, it will become clearer. So what actually happens, how do you get here is, you have ATP, it hydrolyzes to give you ADP, and then a phosphate. This phosphate is actually binding to the substrate. So you have the phosphate going and binding to the glutamate, making a complex called enzyme bound glutamyl phosphate. Right? And then this phosphate is replaced or substituted by ammonia, and that gets releases the phosphate from here, and that finally translates to glutamine. So it's not just a it's not just a single step reaction where ATP is converted to ADP, and the hydrolysis, this energy, whatever is generated from this hydrolysis, is used to drive this reaction. There is a group transfer. This group is first attached, and then this is transferred. That ammonia is exchanged, and that's how it goes and gets connected. Right? So this difference or this information you should always retain. Okay. All right. Now, ATP is not just the only compound that, or not the only molecule that can be used as energy currency. There is PEP, there is phosphocreatine, there is bisphosphoglycerate, there is acetyl coenzyme A. All these compounds which are phosphorylated or have a thioester bond, these are equally very high energy yielding compounds. So if you hydrolyze each of these, or if you ionize these, so hydrolyze all of these compounds, you will get very high free energy change. And these can also be used to drive a reaction. And we do see, right? We do see PEP also being a cofactor for a reaction. So you have PEP, which gives rise to pyruvate, and that pyruvate tautomerises, get stabilized, and because of the stabilization, you have a very high delta free change. Similarly, phosphocreatine gives you creatine, and that again has a very high delta G. Similarly, bisphosphoglycerate and acetyl coenzyme. We will see all these reactions, all these compounds coming up in one or the other pathways as we discuss in the next session. But what you should remember is ATP and all the other phosphorylated compounds and thioesters can serve as energy currency because they have very high delta G when they undergo hydrolysis. And that energy, as we know, drive the reactions. Okay. Okay, so let's see what did we understood so far. So there is set of, I think, 2, 3 questions, and we will understand this concept better as we solve these questions. So here, which of the following factors contribute to large? So we already know ATP undergoes hydrolysis, and it gives a very large and negative free energy change. Now, out of these ones, which one do you think are the reason for that high energy? You first read and then think whatever makes sense to you, and then we will discuss the answers. A, B, C. Okay. Hmm. That's it. Okay. So we are looking at factors that contribute, right? So B and E. Okay. I think B you got right, and someone said A, and both of you said C also. Okay. Let's look into this one by one. Okay, so the first one says that the large free energy change is due to oxygen charge repulsion in adenosine triphosphate. Let me go back to the structure of adenosine, which I purposefully skipped here so that we can discuss this in relation to the questions here. So, sorry, let's look into actually what happens when hydrolysis is going on. What is the but when the hydrolysis takes place, how does the product look like? So this is the adenosine triphosphate. Adenosine, so adenine attached onto ribose. 3-phosphate, 1, 2, 3. So triphosphate, adenosine. This much is correct. Now your hydrolysis, so you're basically breaking these bonds under the so when in an aqueous condition, right? Now hydrolysis relieves of charge repulsion. Where is the charge repulsion taking place? The charge repulsion is actually taking place between phosphate groups. So POP, these POP groups that you see, these these are the one that are repulsion that are facing the repulsion, right? So what happens when you have the hydrolysis, you have this phosphate phosphoanhydride, this one that comes out, right? So this phosphoanhydride is what is given out when there is a hydrolysis reaction taking place. So the repulsion that you know that we know here is, or discharge repulsion is between the phosphate groups and not just the oxygen, because oxygen is not the component that are linked here. The linked components are phosphate. So repulsion is between phosphate. Had it been written of oxygen in phosphate would have made sense, but it's just oxygen, oxygen. That doesn't make sense, right? Now, next, resonance stabilization in the released inorganic phosphate. So let's go back to this again. Now, why should this, we know this, right? We are trying to understand a reaction is favorable or not. It is favorable if the product's formation is uh, favored by stabilization of the product, right? Now, let's look into this. When phosphate actually is out, there is a resonance stabilization here taking place. So here the negative charge, it is distributed over all the multiple oxygen atoms here, and this becomes more stable than ATP. So your this reaction is favorable and it can happen. So the resonance stabilization basically stabilizes the inorganic phosphate that comes out. So that's just correct, and I saw that both of you got that right, so that was great. But if it is resonance stabilization, it cannot be ketoenol tautomerization, because inorganic phosphate is stabilized by resonance. There is no ketoenol tautomerization, at least in inorganic phosphate. It is ketoenol, we know, right? That is CO, which also becomes OH. We have seen this in the previous sessions, but not, there is no such groups in phosphate, right? Next, the D1. This is formation of a stable phosphodiester bond after hydrolysis of adenosine triphosphate. So again, going back to this slide where there is a bond breakage, do you see any additional phosphodiester bond formation when there is hydrolysis? This step here, this is the adenosine triphosphate. Do you see any phosphodiester bond? No, right? The hydrolysis, what happens under hydrolysis is basically breaking of a phosphoanhydrate bond here, but you don't see any additional phosphodiester bond, so that is incorrect. And then E, deprotonation of adenosine diphosphate after the hydrolysis of adenosine triphosphate. That is correct, because here you have ionization and this becomes deprotonated. So this H group was here, this gets deprotonated, making it a ADP 3 minus state. So this is correct. After your formation of ADP from ATP, the ADP, the diphosphate, undergoes a deprotonation. And lastly, conversion of ribose in adenosine triphosphate to deoxyribose for use in DNA, which is again not correct, because you see no such change in the ribose. The ribose, sorry, the ribose is still ribose everywhere. It doesn't suddenly change to deoxyribose. Deoxyribose is sugar that is used in DNA, but that's a completely different thing. It is not part of this pathway or this reaction that we are looking into. So except for the B and the E, everything else is incorrect. And we have seen why. Now this second question, then we move on to the next part. So again, this is again based on the phosphorylated compounds, ATP and several others. And which of these do you think are different? Not difficult. If you read, you should be able to get it easy. You already are able to solve most of the questions, so that's great. B and E, B and E are false statements. Okay. I'll wait for the other person, and if they are commenting, I can see them on time, please. So B, that's it. Okay. So, right. Someone said B and E. That's correct. Someone said only B. That's also correct. The other answer is E. And these are not very difficult to understand. Basically, the other statements are true statements. And we've already seen many of these. The first one said adenosine triphosphate is not the only phosphorylated biomolecule whose hydrolysis is associated with large free energy change. We know, and we know with examples which are the other compounds that are also having large free energy change. So that is correct. Second, the hydrolysis of phosphoryl group in PEP proceeds with a smaller negative free energy change than hydrolysis of ADP. So we have seen this PEP is minus 61.9, whereas this is minus 30. So this is incorrect, right? It is not smaller, it is rather larger. See, the compound obtained along with inorganic phosphate after the individual hydrolysis of PEP and phosphocreatine are each stabilized by keto-enol tautomerism. So if you see here, phosphocreatine gives you a creatine which undergoes resonance stabilization, and it is also, so this is and that is also because of tautomerization. Similarly, pyruvate has enol form, so it undergoes keto-enol tautomerization, right? And then we have D. The compound obtained with inorganic phosphate after the hydrolysis of bisphosphoglycerate is stabilized by resonance, which is here, which is again stabilized by resonance. So resonance stabilization. And E is incorrect. So the free energy change associated with the thioester is greater than associated with the hydrolysis of oxygen ester due to resonance of the former. That is incorrect because oxygen esters are more stable due to resonance, right? So oxygen esters have more have stabilization because of resonance. Thioester lack resonance, and that's why they break down. They have this free energy when they break down. They are not stable anyway. It's oxygen ester which are more stable. Okay, and the sixth one is incorrect, as we know already, because we also know that as the is correct, as the energy currency ATP provides energy by phosphoryl group transfer and not by simple hydrolysis, and we looked into the whole mechanism behind it. All right, let's move into the next part, right? So this you would have seen a lot in chemistry, of course, but also in the lecture notes, where you would have seen that oxygen reduction reactions, not only ATP is used as energy, or not only the phosphorylated compounds are driving the energy or reactions. Oxidation reduction reactions are also one of the important class of reaction that is prevalent in the energy in the cells, right? Now, in oxidation reduction reaction, what is actually happening is there is an electron transfer in such reactions, and that electron transfer, which is the movement of electrons from among the differently due to the electron affinity of the compound, the electron transfer that is responsible for the work done by the living organism, right? So if we are saying electron transfer, or so electron transfer is basically reducing, so the opposite is oxidation, right? This what we understand. So more often than not, you will see that in biological system, if you say oxidation, you are basically referring to dehydrogenation. Dehydrogenation is basically removing of H, which is basically, if a compound is removing H, there is another compound which is accepting H. So that one is getting reduced, so you are basically getting oxidized. So if you are getting dehydrogenated, you are basically getting oxidized. And the class of enzyme that will generally uh, help in these catalyze these reactions are dehydrogenases, right? As the name suggests, dehydrogenases, dehydrogenation they help in dehydrogenation, right? Now, biological oxidation reduction reaction can be described in terms of two half reactions, each with a characteristic standard reduction potential. So, because here we are talking about electron transfer, because here we are talking about compounds getting reduced, so the energy or the potential, how much this compound will get reduced is calculated as standard reduction potential, because you're talking about transfer of electron, right? And whenever there is oxidation, there is a reduction. So there is simultaneous reaction taking place. So there are two half reactions, and those half reactions constitute the overall reaction. So in general, whenever you're looking at biological oxidation reduction reaction, you have these two. You can describe them into half reactions and look at their standard reaction potential. Okay, all right. So let's do this quick exercise, which is very fun also. So before I do, and before I explain it to you, can you give me the oxidation state of carbon, the red colored carbon, in the first three, methane, ethane, and ethene? Let me first ask you this question, and then we will discuss, and then we'll see how do we actually calculate oxidation state. So give me the oxidation state of methane, ethane, and ethene in this figure or in this image here. 876. Okay. Aditi, would you like to answer as well? Give it a try. Methane is 4. Okay. Okay. You're still answering. All right. Seems you're still answering. So maybe wait. Just a second. I will come back to it first. Okay, sorry about the delay. Yeah, so I see you can see answer. Okay, so I see what you are trying to do here. You're trying to find out the oxygen state based on what we know. But when we talk about biological oxygen state, we are talking about if you are sharing, so carbon here, if carbon is sharing electrons with another element, another compound, so another element here, what what happens to the electron pair that is shared, right? So let me give the answer and then we will see why. And I know I can understand if you have any doubt. And someone answered 876, so that person got it absolutely right. And I think that person is already aware of what I'm going to talk now. So in biological oxygen state of carbon, what we are referring when we are talking about this here is if carbon is bonded to H, then or if any other element which is less electronegative, then what happens is the pull of the electron pair is towards the C. So the carbon is basically taking the electron shared electron to on its. So in biological compounds there is unequal sharing of electron between bonded pairs. So if carbon, the red ones here, if you see, if they are bonded to a less negative element, which is H here, then the electron, both the electrons are on carbon. If carbon is bonded to a carbon, then both the electrons are shared equally. So one on each carbon. And if it is with a more electronegative element, which is O, let's say, so both the electron go to O, and the carbon doesn't get to share the electron. So that's the basic concept here. And now let's like to look into 8. What is why exactly 8, and why not the way we calculate generally to give us like the way we calculate. So see, this is the electronegativity order that you need to remember, right? So if C is bonded to H, H is less electronegative. So basically double bond. So the two electrons on each bond here. So there are a total of 8 electrons, and all the electrons will be on C. So 8. Here, let's look at the C in red. So 1, 2, 3, 4, 5, 6. All these 6 electrons will be taken by C, while this electron pair, which is between C and C, will be shared just equally between this C and this C, the black C and the red C. So 1, 2, 3, 4, 5, 6, 7. Again, coming to this one. 1, 2, 3, 4. This is 4 electrons between this C and C. So 2 on each. So 1, 2, 3, 4, 5, 6. Did that make sense? Why we are looking into in this term, did that make sense? And then did the counting make sense? So we are basically trying to understand how many electrons are owned by the carbon. Okay, if that's clear, let's try to do the next 3. Then we'll see, then we can understand if it's more clear. So give me now, with this understanding, the electron oxidation state of ethanol, acetylene, which is base this, sorry, just a second. So this and this, formaldehyde, acetylene and ethanol. This should be easy now. 554. Okay, 5, you type it out, you can check. 5. And about formaldehyde? Fantastic. Both of you got it right. So let's look into it. Ethanol, this red C, okay, 1, 2, 3, 4, 5. This one goes to O, so it is not sharing these electrons, or it is not owning these electrons. Acetylene, this one, 1, 2, 3, 4, 5, 6. 6 electron between C and C, so 3 to each. So 3 and 1, 2, 5. So 3, 4, 5. Formaldehyde, 1, 2, 3, 4. This pair is on, these 4 are on oxygen. So now that I can see you've already done it, so on the right side, which is all these column, acetaldehyde, acetone, formic acid, can you give me the value of these 6? One zero. Oh, sorry. 332 32210. Okay. So I've got one person answering all. The I will wait for the other person. It's easy. It's not at all difficult, and you're already doing so great. So when you finish, then we can discuss the answers. Three, two, two. Okay. Acetonuers. Okay. Formic acid, carbon monoxide is 2, and 2 remaining, acetic acid and carbon dioxide. Mm. Okay. And last, carbon dioxide. Okay. Very well done. Just that acetic acid is 1. So it's 3, 2, 2, 2, and 1, 0. We'll see why. And I think you people got it mostly right, and all are mostly right. So that's great. So let's look into, so remember we are looking only at the red ones. Okay. So acetaldehyde, 1, 2, 3. Acetone, 1, 2. That's why we are not calculating these 4. Formic acid, 1, 2. We are not counting the ones which are shared with oxygen. Carbon monoxide, 2 only, because these are all with oxygen. Acetic acid, we are only considering this one here because we are only looking at this. All these electrons are on O. Okay. And carbon dioxide has nothing because both sides it is with more electronegative oxygen. So the electrons are on oxygen and not on carbon dioxide. Did that acetic acid make sense to you? I think you wrote 7. Yeah, root 2. I understood you took the wrong carbon. But so be mindful. Otherwise, both of you got it right. So that was great. Okay, let's quickly move to the next topic for today. Right. So this image on this towards the right has been there since the start, right? So what does this image here say? This image here is basically talking about how are we measuring the standard reduction potential of a redox pair. The redox pair is basically pair of compounds, one of which is getting oxidized, one of which getting reduced, right? Now, I said, right, there is a standard reduction potential. What is the standard and what is that condition and how do we calculate the potential? So we calculate by this kind of an setup. So what we do here is that we have electrons flowing from the test electrode to the reference electrode or vice versa, right? But what we for ourselves know is the ultimate reference half cell here is the hydrogen electrode, right? And what we know is this is at pH 0. Okay. So the electromotive force, the electromotive proton force or the electromotive force or the EMF, all of these are the same thing. So these force of the electron is basically designated as 0.00 volt. So your EMF is basically designated in the units of volts, right? So we have a test cell which contains 1 molar concentration of the redox pair that we want to examine, right? Now, our reference electrode, we have put as hydrogen electrode, and we are putting that as pH 0. So the electrode has 0 voltage, right? So what we can do is we can do at pH 7. At pH 7 in the test cell, we can put it at pH 7, okay? So we can calculate the electromotive force for the hydrogen electrode. Basically, if this side is pH 0 and we know that this side has no voltage and this side has my redox potential, uh, redox pair working, and my flow of electrons will be in this bridge here via this movement here. And so this machine here, which is calculating the fluctuation in the voltage. So if there is any voltage fluctuation, it's because of the movement of electrons. One side already has 0 voltage. So whatever voltage is because of the movement of electrons. And we can know that movement is from test cell. And if that is against a standard of hydrogen electrode, which is at 0, whatever fluctuation we see is because of the test cell. We are calculating the electromotive force, how much electron is moving and at what extent. Okay, so basically what you need to understand is this is what you do to calculate, but how do you actually calculate? What is the formula? This is the formula that we are using. So we have E, which is the electromotive force or the energy, which is basically just how we did with standard free energy change, right? We had this E, so we have our E naught plus RT and Faraday ln electron acceptor by electron donor. If you look into the delta G formula, you can convince yourself that they look similar, and this is a key for you to remember these both formula. So E naught plus RT ln, the differences will be here, the Faraday constant here, right? Ln acceptor by electron donor. Now R and T can be kept as standard conditions, and then you can simply calculate it as E is equal to E naught plus 0.026 volt by N being the number of electrons in the actual motion, and ln electron acceptor by electron donor, right? This can also be written as, because the energy made available by the spontaneous electron flow is proportional to the change in the energy, right? So you can equate this energy and delta G by minus N Faraday delta E. Okay, or in standard condition, delta G naught by N is equal to minus N Faraday delta E naught. So this formula and this formula You should remember this. You should remember this. I'm pretty sure you, it should be very easy and should come intuitively to you. Okay. So let's make it more easier for us to understand with two numericals that we are going to do right now. So let's do attempt this first one. So you have to calculate the free energy change for this reaction, which is acetaldehyde getting reduced to ethanol. Okay. So your Faraday constant is given 96.5 kilojoule per volt, and your E naught for half reaction. So what is the half reaction here? Acetaldehyde getting ethanol with using 2H, that is one reaction. NAD plus NADH getting reduced. So both of the half reactions are in reduction form. Of course, one of them in the reaction is getting oxidized. But when you are writing half reactions, you are basically reverting the reaction you are writing. So here NADH plus H plus is giving you NAD. So basically if you do it opposite, that is giving you the reduction potential. Sorry, NADH is getting converted to NAD. So NADH plus H is basically how it is reducing it. So you are basically, when you are writing half reactions, you are writing for both the reduction reactions, and you have the E naught for that, and you have Faraday constant with you. Calculate the free energy change. If it doesn't make sense, we'll do it. But if you have looked into the notes, you would be able to do it. Point 934 Faraday. Well, I am asking you in terms of delta G. So could you calculate in delta G for me and let me know the value? The question asks calculate the free energy change, which is basically delta G. So basically we are supposed to use this formula here. So you can, so you know this F is already given to you. We can guess the N, or we can understand what is N from the equation that is given, and delta E we need to calculate. Okay, maybe one more minute and then we can discuss. You can look in the answers. It's not very difficult. I can understand where the confusion is arising, but it's okay. Let's just give it a try. Okay, maybe I will show it to you because I think I can understand where the confusion is drawing from. Right. So I showed you this formula already, right? This is what we are going to use. Delta G naught is equal to minus N Faraday delta E naught. So where do you get the delta E naught for a reaction like this? So how do you express your delta E naught in a reaction when you are given a reaction? You basically calculate E naught of electron acceptor minus E naught of electron donor. Make sense? So delta E or reduction potential difference of the reaction that you are given is calculated by electron potential reduction potential of electron acceptor minus electron potential of electron donor. Right? A reduction potential of electron acceptor. What is the electron acceptor here? And let's aldehyde because that's getting reduced to ethanol. So you have minus 0.197 volt, which is subtracted with the electron donors reduction potential, which is also given to you minus 3.320. You basically just do the calculation, come to 0.123 volt. Then I said that this part is now calculated and you need to find out. And you can see in the equation, you can see 2 electrons, 2 hydrogens. So 2, number of electrons is 2. You have 2, you have Faraday constant given to you already, and you can already convert, use 0.123 from here, and that is the answer that you have, minus 23.7 kilojoule per mole. Did that make sense? Any confusion? What you need to remember is how to calculate it. So I wanted to show this, and that's why I didn't want it to be in the text form. I wanted it to be such that you already see the example and get to do it. Okay. Yeah, sorry. In the first case, you have calculated delta E. Yes. I'm losing it. Then, sorry. So you have to calculate it in terms of delta G. Then you can convert it to delta E. I didn't understand. You have to convert it into terms of delta G, and then you convert it in terms of delta E. Where do you do that? I'm sorry, I must be missing it a bit. So I go equation here maybe. So okay, let me understand this better. Or maybe I'll do one thing because I don't want to take up others' time. I will come back to this question by leaving you on this question, and then I can go back and look into the what the doubt was. So we have already done a part of this question. These are the standard conditions. Now, the same question, however, I have changed the concentration of the products and the reactants, right? You now have to calculate the delta free energy change for me. Okay. So the same question, same values, Just that right now, my products and my reactants concentrations have changed. So can you calculate the phase energy change while I am looking into the doubt posted in the chat box? I'll leave you with this question.
First case you have in the first case you have calculated the delta E, but delta is not J in terms of delta G, and then you can convert it to delta E.
Okay.
Just a second.
Okay. Uh, yeah. So while you are answering, okay, maybe I will don't want to take another time into this. So when I am done answering this question, then I'll come back to this question, the doubt posted in the chat box, and I can have a discussion on that. Okay. So first we'll try to solve this question here, and then we can see what the doubt says here. Okay. Okay. Is this question visible to people? Is it difficult? It shouldn't be that difficult. Okay, one more minute and then we solve this question first and then we look into the answer. May I show this as the answer? What we have done here. Yes, it might look very confusing. Let me be a little easier on this. Okay. So the concentration of acetyl and NADH are present at 1 mole. So you have the concentration at the beginning. And then ethanol and NAD. So you have the reactants concentration and the product concentration. But remember, these are half cells. So you need to calculate the potential for both these reactions. So when such kind of concentration is given, because the concentration is incorporated into the reduction reaction. So let's say acetyl getting reduced to ethanol. So that is one reaction where you have acetaldehyde and ethanol concentrations. You first calculate the E-acetaldehyde potential for this and potential for NADH, and then do the delta D that we have just used. And then you calculate delta G, just how we have done in the previous question. But the delta G, delta E here, which is basically the acceptor minus donor, that will first come from you incorporating the values of products and reactants in the equation here. Okay. So when you do that, you have minus 0.197 plus 0.026 because these are standard conditions by 2 because you know this already, 1 and 0.1, 1 and 0.1. Same thing we have done and we have got minus 0.167 and minus 0.350. We did the same calculation that we did in the previous question, minus 0.167 minus 0.350, which is 0.183. And then we calculated the delta G, right? So these two questions, yes, I understand why it would have looked a bit difficult, but yeah, so that makes that component here. Yes, 35.3. I can see Dipali has get this already. Is Priyabrata still here? Yeah, okay. So Priyabrata, coming to your doubt here, which you said that delta E is not additive. Well, that is the case, but the question, the example that I have taken is from Leninger. So you can do the reading in Leninger additionally if I am not able to clear your doubt. But what I understood is when you are looking into the potential difference, delta E, which is basically the difference between the two reduction potential that is taking place, you're basically saying what is the how much is the electron flow from the donor to the acceptor, right? And we are computing the reduction potential uh, from the donor to the acceptor, because acceptor of course has greater tendency to accept electrons, so we are doing acceptor minus donor. Now this delta E is generally positive. Okay, and you can calculate that, and that's why you are able to put it with delta G, because delta G, so basically if you have delta E as positive, you have a spontaneous reaction. We did this in the beginning of today's session, so we know it's a spontaneous reaction and we know delta G can be equated in that way, right? So basically what we are trying to say here is, or maybe I'm not understanding the question properly, because we are not adding anything here. We are looking at two electrodes in the same cell. We are trying to find out the potential difference of the cell where electron transfer has taken place. These are not two in two, what do I say, um two independent steps that are taking place. One is getting reduced. These are concomitant with each other. One is getting reduced, one is getting oxidized. So when you're calculating the potential difference, you're basically just calculating which one has greater tendency and which one drew more electron from the other. And that's why you're adding the delta E here. And then when you have the delta E, and you know this is a spontaneous reaction because this has a greater tendency of pulling electron, this will happen spontaneously. So you have that delta E, and you know that this can take place in thermodynamically. This is favorable. So you can then equate it with delta G. That is what is happening here. If you are still not clear, please let me know. Please, you can type your question in the chat box, because that's what I understood. That because it is a spontaneous reaction that can take place. Just a second. Yeah, because it's spontaneous in its own nature, there is no separate step here. One is not dependent on the product of the other. It's both concomitantly taking place. Both are taking place together because one is more electron accepting than the other. So it is able to withdraw more electrons from the other. So this is a spontaneous reaction on its own. And then this is equated with delta G, right? Okay. Yeah. So that was the answer to the previous question. And I see we are only 13 minutes left. That's okay. We'll try to solve this question. And after this, if we have time, we'll move into glycolysis, or we can do glycolysis completely as a separate topic in the next session. Today we had the first 5 steps, but I don't think so we can complete all the 5 steps. So if we are less on time, then we will do glycolysis from the start to the end in the next session. We will just wrap up with this question today. So basically here you have a formula given. So when the pH probe is dipped into the sample, potential difference between the internal electrode and the reference electron will change. And this you have the Nernst equation here. With the equation that we have been using so far, it's called the Nernst equation. You have E reference minus E sample given by this formula that we have been so far using. Now the question here is, if a buffer of pH 7 is to be made, what should the absolute value of potential difference in volts between the two electrodes? You have all the necessary values given to you. Temperature is given, reference potential is given, R is given, F is given, and the ln10 is also given to you. This one is pretty straightforward, so this shouldn't be an issue. 0.16. Okay. Do you want to revise your answer once again? Maybe just, if you have taken care of the signs and the symbols, just check once again maybe. 0.16. Is it 16?
We have all the values already given to us, so it shouldn't be a very difficult thing to do. Your temperature is given, your reference potential is given. Mm, yes, yes, that's correct. That's correct. Since pH is log minus log H plus at pH 7, we get 10 to the power minus 7. That's also correct. But where do you use it then? You have a formula to give given to you. So you can, so I understand you put this value there and you've written 0.16. So I just want to nudge you there. I just 0.16, or is there a value difference to it? You've got the values correct. You can see the formula. Maybe I'll show the formula once again. Let's say this is the formula, right? Or maybe, wait a minute. Where is the formula? The formula is here also, right? So you have ln H plus. So your H plus is there. Ln H plus is what you need to find. And let's say you do that also, and then you need to substitute that in the formula. Minus 0.41. Yes. Yes. Minus 0.413. Okay, maybe 1 minute more for others to also give me the answers if you have reached to this part, and then we can look into the answer. And I can see that we will have no time left for discussing glycolysis, which is very interesting topic and very intrinsically it's written like the whole pathway is very intrinsically described, and if you understand that, I'm sure you'll never forget. So I don't want to just describe it very quickly in the first 5 step in very quick manner. So we'll do that entire thing next session completely. We'll look into it broadly and in with more descriptive way. So today, after this question, we can stop because we already are about to over. E sample would be 0.663. Yeah. So we have two values, and the exact way that Priyabhadra has written is exactly how we're going to discuss. So you give me the E sample and then the absolute difference also that you have already given me. So yeah, I will wait if people are, because it's very easy. It's not very difficult. I understand the logarithmic and the ln values difference. So I know it. If people reach there, they will be able to solve it. So I will just wait for 1 more minute if other can come to that answer, and then we can wrap this up. Okay. Okay. I will give the answers. So I've given you the E sample value, but potential difference is also, sorry, the absolute potential difference is also what we're going to discuss when we're looking at the answer. So yeah, okay. This is the formula that we are referring, right? It's already from the question. The question says E reference minus E sample is equal to RT by F ln H plus. On the left, you have all the values that are given here. The temperature in Kelvin, you should take. So it's in Kelvin here. Reference potential as it is.
R as it is because it's a constant. F, Faraday constant, is a constant. Ln10 is already given to you. That's why that information is incorporated here. And pH is 7 is given to you. So pH 7 means H plus is 10 to the power minus 7. This much you should remember. This much already someone had put in the chat box. Now, what I generally also do is I divide my entire equation into multiple parts so that's easier for me to work. So this is what I have as a result also. So your RT by F, which is all the constant and values that are not changing at one place. So I have calculated this portion RT by F first. So 8.3149650 divided by 9650 and again into 2298. I get a value of 0.0257. This much is okay. This much everyone will get, I'm pretty sure, because it's not changing. Now you have ln h plus. So ln 10 to the power minus 7. If you simply solve it, you get minus 7 into 10 to ln 10. ln 10, that's why ln 10 is given because that's how you can open it, right? So you ln, when you do that ln 10, it's 2.303 into minus 7. That is minus 16.124. Right? So RTF into ln H, which is this into this, is minus 0.414. This is what people got. 0.16. Dipali, I'm still not sure why are you getting 0.16 and where is the 16 from? Are you using something else? Maybe just check, maybe in points and just decimals, you can check where you're getting that correct, the value. So this is what people wrote, minus 0.4 and 4, because I asked for the difference between reference and sample. But you need to also report your, so absolute potential difference is actually the E sample. And for that, how do we know that we are supposed to use it? We already given reference potential. So you have 0.250 and you just calculated the factor here, which is 0.414. So if you add them up, it becomes 0.663. So that's the answer. Dipali, are you still having 0.16 or did you manage? Did you understand where you are not getting the 0.4 or 0.414 from? Can you let me know where the difference is or what exactly you are facing problem in? Maybe. Yeah, and anyone else, if has any doubt, please let me know. I will be happy to take them. And if I can't help you with that, I will definitely try my best to come back with answer. Yes. Yes, pretty much yes. I agree with what you have said, Priyavrata. You can do it simply like that. Why I chose or why you would see people doing in this way is they basically try to make use of all the information here, right? You did the same thing. You did minus 2.03 RTF. We are doing the same thing. We are it becomes comfortable with. This is any other. There are multiple. How do you say? Uh, multiple equations that lead up to the same concept. You use any of the equation and reach up to the answer. That's important. If you have that understanding, and I am glad that you can think of other possible equations, and that's fantastic. Please do keep it up. Just that this step is also not, it's complicated, yes, but it also gives a way to for you to discern at every step what is exactly happening. I was studying this equation. How do I put this equation here? That's also a reason we did this. But at the same time, I would say that this is perfectly normal if you are reaching the same answer. But then you would not as it would be apparent in this procedure when we're following. But that said, if the answer overall for the E not sample is 0.66, that you can reach by this also. Right. But if anyone else has any doubt, also please mention. Otherwise, we are done for the day. If you have any doubt, please mention. If you have any question, please do let me know. Otherwise, we meet each other in the next session where we discuss about glycolysis. Thank you so much. Thank you. Thank you. Thank you. Yes, please. Thank you. If you have no doubt, please we feel free to exit.